Hello everyone. Welcome to the new session of Anatomy of Flowering Plants exercise. So in this session, we are going to study about the anatomy of flowering plants. Okay. And we are going to discuss the NCERT questions over here. So here comes the first question. Draw illustrations to bring out the anatomical difference between monocot root and dicot root. So first question is here. You have to represent the difference between, okay, monocot and dicot root on the basis of their anatomical structure, right? And the second question in the same, that is the B, monocot stem and dicot stem. So again, you have to elaborate the differences on the basis of monocot stem and dicot stem on the basis of their anatomical structure. So let us see here. So here you can see the monocot root and dicot root. If you clearly see the structure, then what you see, their outer surface, that is the presence of the epidermis, and here the epidermis is called as epiblema, whether it is a monocot or dicot, and it gives rise the unicellular root hair. So here you can see the epiblema. The root hairs are the growth of the epiblema cells. So here you can see the root hairs coming out from the epiblema cells. Now, just below this, you can see the this hypodermis structure. So here the cells are very compact in both the cases, right? And then you can see the anatomical structure, the other thing, that is the cortex region. So there is a cortex region, okay? And below the cortex, okay, there is an endodermis cells. So in both the cases, you can see the endodermis cells. The endodermis cells in the case of monocot root, here you can see just a single layer over here. But in the case of dicot, here you can see the cells are of two to 3 layers, right? Here you can see the 2 layers in the case of this dicot root, endodermis layer, right? And below this, there is a pericycle. Just below the endodermis, there is a pericycle in the case of dicot root. But here in the case of monocot root, you won't see any pericycle, right? There is no pericycle, you can see here. Okay, so this is one of the major difference on the basis of this endodermis and pericycle. Endodermis in the case of monocot root, it is just a single layer, and here 2 layers you can see in the endodermis. And on the basis of the pericycle, you can see the pericycle is not having any layer in the case of monocot root, whereas in the case of dicot root, there is a pericycle layer, right? Okay, now coming to the vascular bundle. If you see the vascular bundles in the case of monocot, the xylem and phloem are at the radial. Okay, arrangement is the radial. So both the xylem and phloem are on the different radius. So here you can see the radius, right? At this xylem is present. On the another radius, you can see the phloem is present. So they are not a conjoint, right? They are not conjoint. And like this, you can see the number of, okay, the number of vascular bundle is more. Okay, the number of vascular bundle in the case of monocot root is more if you compare it with the dicot root. The dicot root, the vascular bundles, you can see just 4 xylems alternate with the phloem. So here also you can see the xylem and phloems are on the different radii, right? They are present on the different radii. Now, if you see after this, you can see the protoxylem and the metaxylem. Now, the xylem is more, you can say, having the layers, okay? In the case of dicot, you can see 1, 2, 3, 4, 5 different cells, and they are having the metaxylem toward the inner side and the protoxylem towards the outer side. Same here, the arrangement, you can see the metaxylem is inner side and the protoxylem is again the inner side. But if you see the pith in the case of monocot is well grown and it is enlarged, right? The pith is more, right? But what about the pith in the case of dicot root? The dicot root is having very small pith, right? So these are some of the difference on the basis of the anatomical structure in the monocot root and dicot root. So let us see now the difference on the basis of anatomical structure in the case of roots of monocot and the dicot stem. Monocot stem and dicot stem. So here you can see the monocot stem. Okay, the monocot is having there is vascular bundles scattered all over the cell, right? So there is a epidermis, there is a hypodermis, right? And just below the hypodermis there is a scattered vascular bundle. This is a very unique type of structure you can see in the case of monocot stem. Okay, there is no endodermis, there is no pericycle, and the vascular bundles are scattered. Now if you see the diagrammatic structure of the dicot stem, what you find here, there is a epidermal hairs, and this epidermal hair is not present in the case of monocot stem. No epidermal hairs are present, right? And here you can see the epidermal hairs, that is also known as trichomes over the surface, and that is the outgrowth of the epidermis, right? Now below the epidermis you can see there is a collenchyma cells. Okay, collenchyma, several layers of the collenchyma cells, and after that, and that makes the hypodermis, right? Below that there is a parenchyma cells that makes the cortex region, right? And you can see here the layer of endodermis and many layers of pericycle. The pericycle is well grown in the case of dicot stem, and that is totally absent in the case of this endodermis, and pericycle is totally absent in the case of monocot stem, right? Now, if you see the vascular bundle, so the vascular bundle here you can see that is the conjoint type, right? The xylem and phloem, and in between you can see the cambium layer. Very, very important. It means that open type of vascular bundle is there, right? So cambium is present in between the phloem and xylem. And here you can see there is conjoint vascular bundle, xylem and phloems are present, but there is no cambium. Cambium is totally absent. So closed type of vascular bundle is there. No secondary growth can be seen in the case of monocot stem, whereas in the case of dicot stem you can see a well growth of the secondary, okay, secondary xylem and phloem, right? Because the cambium is there, right? And also the pericycle is there, so that can divide and redivide and forms the lateral, okay? The lateral, you can say the bark can be synthesized from this pericycle if there is a secondary growth is here. So here you can see the difference between the monocot stem and the dicot stem, okay? And that is totally based on the anatomical structures. Now, the next question comes here. Cut a transverse section of your stem of a plant from your school garden and observe it under the microscope. How would you ascertain whether it is a monocot stem or the dicot stem? Very important question. It is just based on your activity that you have to cut any plant, okay, transverse section of it and visualize it under the light microscope, and you have to tell that whether this stem which you have taken the slice of it is a monocot stem or the dicot stem, okay? And you have to give reason how you can say this a monocot or a dicot stem, okay? So let us see the answer of this. The following characteristics can be used to distinguish it is a monocot or a dicot. So how can you say that this particular structure is of dicot stem? Vascular bundles are arranged in rings. If the vascular bundles are arranged in ring, then you can say it is a dicot stem. Okay, it is not scattered. In the case of monocot, you can see the vascular bundles are totally scattered, right? In the monocot stem, right? But here you can see the vascular bundles are arranged in a ring-like structure, ring form. Okay. Vascular bundles are open. Open means the cambium is present. If the cambium is present, then you can say that this is a dicot stem. And in the case of monocot, they have closed vascular bundle with bundle sheath. So there is no presence of cambium, right? So this is a very unique difference between the dicot stem and the monocot. So these structure you can see under the microscope, and just by seeing these structure you can tell that whether the stem, the slice, okay, the section which you have cut from your garden stem, where it is a dicot stem or a monocot stem. Okay. Presence of the collateral, conjoint and open vascular bundle. Now, if you see the clearly each and every vascular bundle, then in the case of dicot stem, what you see, if it is a collateral vascular bundle, conjoint means xylem and phloem are present at the same radius. It is not present on the different radius. It is present on the same radius. And open vascular bundle. Okay, cambium is present with the strip of the cambium between the xylem and phloem. Open means the presence of the cambium. Okay, the strip of cambium. Now, if you see a single vascular bundle, then what type of vascular bundle you see in the case of monocot stem? Presence of collateral, conjoint and closed vascular bundle. Here it is again the collateral and conjoint type of vascular bundle, but the vascular bundle is closed type. It means that it is not having any cambium layer, right? So closed vascular bundle dispersed in the ground tissue that contain the parenchyma. So they are containing the parenchymal cell. Okay, so this is the difference between the dicot stem and the monocot stem. Ground tissue can be differentiated into the parenchyma, collenchyma and endodermis, pith and pericycle. So this is a very unique thing that you can see in the case of dicot stem. When you see the ground tissue, so ground tissue can be differentiated into the parenchyma. Okay, so the most just below the epidermis you can see the collenchyma cell. Okay, that is the hypodermis cells in the case of dicot, and later after the 2-3 layers of the collenchyma, then you can see the parenchyma layers. Okay, there is also a presence of endodermis, pith and pericycle. So these layers you can easily find in the case of dicot stem. Medullary rays are found between the vascular bundle. You can also see the medullary rays present in between the vascular bundle, right? So there are the parenchymatous cells which forms the medullary rays, and that is present in between the vascular bundle. But what about the monocot stem? Phloem parenchyma is absent. There is no phloem parenchyma. This is about the vascular bundle, and water containing cavities are also present. Okay. So here you can see the monocot stem and the dicot stem. Okay. The vascular bundles are present radially. You can see they are present in the ring form, but in the case of monocot stem, they are scattered all over the this stem region. Okay, the ground tissue is not differentiated into the collenchyma, parenchyma, endodermis and pericycle, right? Just below the epidermis there is the epidermis layer, there is the hypodermis layer, and you can see the vascular bundles are scattered all over. But here what you see in the case of ground tissue, there is an epidermis, there is a hypodermis made up of the collenchyma cells, then there is several layers of the parenchyma. Okay, then comes the endodermis and then comes the pericycle. So these layers are totally absent in the case of monocot stem, right? Okay, so here comes the next question. The transverse section of a plant material shows the following anatomical features. Okay, so here we can see some of the anatomical features. What are those? The vascular bundles are conjoined, scattered and surrounded by the sclerenchymatous bundle sheath. And B, the phloem parenchyma is absent. And what will you identify it as? So the transverse section, when you see the transverse section, you see such of the following anatomical features, right? So the transverse section is of monocot stem. If you see such type of structure, then what you can see that it is a transverse section of monocot stem, because the vascular bundles are dispersed in the monocot stem. In the previous questions only we have seen that vascular bundles are scattered, right, all over the stem region. There is no collenchyma cells, there is no parenchyma cells, there is no endodermis, there is no pericycle, right? And this vascular bundles are scattered all over the stem. So the phloem parenchyma, and when you see this vascular bundle, a single vascular bundle, then in the case of phloem parenchyma, this vascular bundle, the phloem parenchyma is absent in the case of monocot stem. So this shows that the transverse section which you are visualizing under the microscope with such a feature, it is of a monocot stem. Okay. Okay, here comes the next question. What is a stomatal apparatus? Very, very important question. What do you understand by, in fact, a stomatal apparatus? Explain the structure of a stomata with a label diagram. So with the help of label diagram, you have to explain the stomatal apparatus. So let us see what is a stomatal apparatus. Stomata are the structures present in the epidermis of the leaves. Okay, stomata are the structures. We all know that the stomata are the modification. In fact, it is the modification of the epidermal cells. So they are confined at the epidermis layer only, right? And the stomata regulate the process of transpiration and gaseous exchange. So what is the function? Function of stomata is to regulate the process of transpiration and gaseous exchange. It means that when the stomata is open, there is a gaseous exchange. For gaseous exchange for the two life processes, that is the photosynthesis and the respiration. And also they are working for the transpiration. It means that whatever the extra water is present in the plant, it vaporize and transpire, right? So there is a suction pump which helps to absorb the water. So that is another thing. We are not going to discuss about the transpiration, but yes, the stomata regulate the process of transpiration and gaseous exchange. Now each stomata is composed of two bean-shaped cells. When you see a single stoma structure, what you see, the shape of the cell, that is the stomata structure, it is made up of bean shaped, right? Two bean shaped cells, when come close together, it form an aperture, okay, opening, that is called as the stoma, right? And these bean shaped cells are called as guard cells. When you see the cells of this stomata, okay, then you can see that they are in bean shaped, and those cells are known as guard cells, which encloses the stomatal pores. So they are the cells that sometimes the stomata is open and sometimes the stomata is closed. So these two cells are there to regulate the opening and closing of the stomata. Now the guard cells are dumbbell shaped, where their outer wall is thin. Okay, sometimes you can see in the case of grasses, the shape of the guard cell is like a dumbbell, right? So where their outer wall is thin, generally you see this cells are in the shape of the bean shape, but in the case of grasses, they are in the guard cells. And there, if you see the outer wall of the stomata, it is very, very thin, and the inner wall is highly thickened. So this thickened wall is there to regulate the opening and the closing of the stomata, right? So the stomata structures are very, very important. It is there to regulate the opening and closing of a stomata. Now, these structure possesses chloroplast and regulate the closing and opening of the stomata. So this guard cells are having a chloroplast in them, and it means that they are doing the process of photosynthesis. They are synthesizing the sugar, right? They are synthesizing the food material, glucose, right? And that can regulate the opening and closing of the stomata. The epidermal cells near the guard cells in some cases become specialized in their structure, shape, and size. But what about the other cells? These we are talking about just the two cells that is making the guard cells. But what about the other cells? The epidermal cells near the guard cells in some cases become specialized in their structure, shape, and size. Some of the cells, they get modified, they get specialized in their structure, in their shape, in their size, right? And they are referred to as subsidiary cells. Very, very important. The cells that is surrounding the guard cells, right? They are specialized in their structure, shape, and size, and they are now called as subsidiary cells. The guard cells, the stomatal aperture, and the girdling, means surrounding this guard cell, that is subsidiary cells, are collectively referred to as the stomatal apparatus. Okay, so three things are there. You have to remember what is making the stomatal apparatus. Three things are there: the guard cells, the stomatal aperture, the girdling subsidiary cells, and that are totally collectively referred to as the stomatal apparatus. Okay, so here you can see in the figure the stomata with bean-shaped guard cells. So when the guard cells are in the bean shape, you can see this is the bean shape. The outer wall of the guard cell is very thin, whereas in the case of the inner wall, you can see it is highly thickened, right? And you can see the chloroplast in the case of guard cells, right? Now, what about the other cells surrounding the guard cell? You can see these subsidiary cells. They are modified. Okay, they are not ordinary epidermal cells. They are modified, and you can see there are a number of subsidiary cells surrounding this stomata. Okay, surrounding this guard cell, in fact. So there you can see 1, 2, 3, 4. 4 guards here, subsidiary cells are there surrounding the guard cells. Okay. Now, what is then this apparatus, stomatal apparatus? This guard cells, the stomatal pore or the aperture, that is the opening. Okay, that structure, guard cells and subsidiary cells unitedly called as stomatal apparatus. Okay, so this is about the bean-shaped cells. Now here you can see the dumbbell-shaped guard cells. Dumbbell-shaped. So here you can see this the hand region where you hold the dumbbell, that part. So that is highly thickened, whereas the other part, the bulging part outside, it's not so thickened. Okay, and you can see the two subsidiary cells, which are very, very large cells surrounding the two guard cells. They are the subsidiary cells, and you can see the stomatal aperture is also there. So here also the stomatal apparatus is made up of the three things: the guard cells, the stomatal aperture, and the subsidiary cells. Whether it is a bean-shaped cells or a dumbbell-shaped, the stomatal apparatus is made up of the three things only, right? So in both the cases, you can now see what is the stomatal aperture and stomatal subsidiary cells and the guard cells that unitedly make the stomatal apparatus. Okay. Now comes the next question. Name the three basic tissues system in the flowering plant. So we have seen the three basic system of tissue. Okay, basic tissue system in the case of flowering plant. Give the tissue name under each system. You have to name the tissue under each system. So what are the three that system? So following are the three basic tissue system in the flowering plants. In the flowering plant, you can see the three basic system of the tissue are the number one, epidermal tissue system. So this epidermis is present all over the plant surface, whether it is a stem, whether it is a root, whether it is a leaf. Okay, the outer surface of the flowering plant, any organ, whether it is a stem, roots and leaves, there is a outer surface that is made up of epidermal cells, right? And it forms a tissue, and that particular system is called as an epidermal tissue. So let us see what confine this epidermal tissue system. The epidermal tissue system include the epidermis and epidermal appendages. So there are many epidermal appendages that comes out from the outer surface of the epidermis. So that is also included in this epidermal tissue system, right? So there is an epidermis and epidermal appendages. Now, epidermis comprises epidermal cells and the guard cells. Now, we all know that this stomata, which is present on the surface of the epidermis, okay, it is at the surface only. And this stomata is modification of the epidermis. It is the modification of the epidermis. Okay, the epidermis comprises epidermal cells and the guard cells, and those guard cells are modified epidermal cells only. Now, with the epidermal appendages include root hairs. When you are talking about the root, then also there are some epidermal appendages. So that is root hairs. Now, you can see the epidermal appendages on the surface of the stem also. So that is stem hairs. There is also the stinging hairs and the glandular hairs also. Some of the hairs are sometimes having the glands in them, and they secrete some exudates, right? Some liquid substances. So there are the glandular hairs, there are the stinging hair, there are the stem hairs on the surface of the stem, and in the root surface, now you can see there are some root hairs also. So all these hair like structure, whether it is present on the surface of the root or the stem, they are the modification of the epidermis, or you can say that they are epidermal appendages. So they are the part of the epidermal tissue system. Okay. Next is the ground tissue system. So what we see in the ground tissue system, the ground tissue system is made up of simple tissue, very simple tissue, right? So there are three different types of ground tissue: the parenchyma, the collenchyma, and the sclerenchyma. Okay, so we have already discussed about this, but I just want to here focus on the parenchyma, collenchyma, and sclerenchyma cells. So let us see what are the parenchyma cells. Parenchyma cells are the isodiametric cells, which is having a very thin outer membrane, or the cell wall, which is a thin cell wall, and it is having a large vacuole in the center. So the nucleus is shifted towards the periphery. So this is the arrangement of the cellular structure, and you can see there are many intercellular spaces in the case of parenchyma cells. Okay, the parenchyma cells are having the intercellular spaces in between. And when you see the collenchyma cells, the outer wall is quite thickened, and at the corners, you can see the thickening. There is a deposition of the chemical substances, and the intercellular spaces are reduced. Okay, so this is the difference between the parenchyma and the collenchyma. Now this parenchyma is differentiated, or you can say the modifications of the parenchyma can be seen, and there are the two main modifications. That is the parenchyma, sometimes they are called as chlorenchyma. So when they are called as chlorenchyma, when they are having the chloroplast in them, the organelle that is involved in the process of photosynthesis. And that is why it is called as chlorenchyma, because they are having the chloroplast. Inside the chloroplast, they are having the chlorophyll, right? Now, there is another modification in the case of aquatic plant. This parenchyma, it's having a large air cavities in between, okay? And that is called as aerenchyma. So the air get filled in these cavities, make the plant very light, and the plant can float on the surface of water. So this we see in the case of modification of the parenchyma. We have talked about the collenchyma, now the sclerenchyma. So this sclerenchyma is a fibrous tissue. The internal protoplasm is dried. Only the cell wall is there. Okay, so they are basically the dead tissue. right? This sclerenchyma is the dead tissue, but the parenchyma and collenchyma are the living tissue. They are having the protoplasm structure in the cell, so they are living in nature, but this sclerenchyma is dead cells. Okay, so the main function of this sclerenchyma is to provide the strength and you can say the stability, right? And the parenchyma, we already talked about that. Parenchyma is having the dual function. Sometimes they store the food material, sometimes they are having the chloroplast in them. Then they can do the process of photosynthesis, and in some aquatic plants they are modified for as a parenchyma also. So they provide the buoyancy to the plant, and the plant can float on the surface of water. So that is because of the parenchyma. Collenchyma, they provide the flexibility to the cells. Okay, they are very flexible. So this collenchyma provide the flexibility in the plants. So this is about the second ground tissue system. Now we will talk about the vascular tissue system. Vascular tissue system consists of the complex tissue such as the xylem, phloem, and the vascular cambium. So this vascular tissue, in the case of vascular tissue system, we see that there are two different types of the complex tissue. Okay, these are called as a complex tissue because many different types of cells unite together to perform a one particular function. So this is showing the complex type of tissue. They are not one single type of cells and united to form the tissue. There are different types of cells. In the case of xylem, we are having the four different types of cells. There is a xylem vessels, xylem tracheids, xylem parenchyma, and xylem fibers. So four different types of cells, and out of these only xylem parenchyma is living. Rest other three are dead in nature. And its basic function is to transport the water and mineral dissolved in it. And this is showing the unidirectional movement. What about the phloem? Those phloems are again, they are also made up of four different types of cells. Sieve tubes, companion cells, phloem fibers, and phloem parenchyma. So just leaving this phloem fibers, all the three are living. Phloem fiber is dead, whereas the other cells are living in nature, and they are meant for the transportation of the food material. The food material which has been synthesized with the process of photosynthesis. So they are used, means they are involved in the transportation of those food material, right? And they are showing the dual bidirectional movement. Okay, so the food material can move from upward to downward and downward to upward as well. Now, vascular cambium, in this between the xylem and phloem, when the vascular bundles are open type, they are having the stretch of cells that is called as a cambium. Two, sometimes one layer, sometimes two layer, and they divide and redivide and forms the secondary xylem and secondary phloem cells. Okay. So that is about the secondary growth. So we have seen these three different types of tissue system: epidermal tissue system, ground tissue system, and vascular tissue system. Okay. Next question. How is the study of plant anatomy useful to us? Why we are studying this plant anatomy as a chapter, right? In our curriculum, in your syllabus, this anatomy of flowering plant is there. So what is the use of this chapter? Why we are studying this chapter? The question is about that. So the study of plant anatomy is useful to us in the following ways. So this study, the plant anatomy is very, very important, very useful in study. Why? Because it helps us to understand the structural adaptation in plants to different climatic conditions. So you can see there are so many modifications on the basis of their anatomical structures in the case of plant, and those adaptations are due to because they are living in the different environmental condition, different climatic condition, so they have to adapt according to that. So there are so many modifications on their anatomical structure, right? So that is why it is very, very important to understand how they are getting adapted to the different climatic conditions. Second point, helpful in identifying the monocot, dicot, and gymnosperms. So if you know the anatomy, right, you can see the internal structure of the plant, and you can say that this particular plant is the anatomical monocot plant. If you see the different type of anatomical structure, as you see in the case of dicots, stem or root, you can say that this is the dicot plant. And same with the gymnosperms, because this type of plant means monocot, dicot, and gymnosperm, they are showing the different type of anatomical structure. So it is very helpful in their identification, in their classification, in their taxonomy, right? So nowadays we are considering the anatomical structure in their taxonomy study also, right? So it is very, very important in that way. Physiological condition can be studied which help in the crop improvement, right? So we can see there are so many different adaptation to the physiological conditions, because their plants are growing in the different environmental condition. So they are we see there is a modification in the physiology also, right? So this also help us to understand to how the this anatomical structures are different and why they are different on the basis of the physiological activity, right? And this will going to help in your crop improvement. Next point is the study of the plant fibers such as flax, jute, etc. Because these fibers are very, very important. They are used on the commercial basis, right? So they are helps in their commercial exploitation, as it enables the predictions of the strength of the wood, which can be utilized to its potential. So by knowing the anatomical structure, you can tell that this particular plant is going to provide this type of fibers, and it is having such type of adaptation, such type of characteristics, and this can be used for further different process. Okay. Next question, describe the internal structure of the dorsiventral leaf with the help of labeled diagram. Okay, so here you can say that you have to find out what is the internal structure on the dorsiventral leaf. So first of all, we have to understand what is the dorsiventral leaf. Dorsiventral leaf means the leaf which is flat, and it is they are having the dorsal surface, they are having a ventral surface, right? So let us see. Dicot exhibit the dorsiventral type of leaf. Okay, if you see the dicot leaf, then they are showing the dorsal surface and the ventral surface. So that is why the leaf of the dicot are called as dorsiventral leaves. Okay. When examined, the vertical section of the dorsiventral leaf, it consists of three different parts, and they are, so when you see the anatomical structure of the dorsiventral leaf, you can see the three different internal structure, three different parts. And what are the three different parts? Epidermis, the number one point, that is, you see the epidermis layer. Epidermis is present all over the plant surface, right? So it is present on the leaf also. It is found on the adaxial epidermis, that is the upper surface. Okay, so there is an upper epidermis and there is again the lower epidermis. So lower surface or the lower epidermis is also called as abaxial epidermis. Abaxial epidermis. Okay, now on the outside, the epidermis is covered with a thick cuticle. If you see the surface of this epidermis, you can see the outside, that is the epidermis, is covered with a thick cuticle layer, very thick cuticle layer, right? And as compared to the upper surface, the abaxial epidermis comprises more stomata. Okay, so the lower epidermis, the abaxial epidermis, is having more stomata as compared to the upper surface. And you can answer this, why they are having the more stomata on the lower surface than the upper surface. Just to minimize the rate of transpiration. So because the sunlight is coming directly to the upper surface, but it is not directly reaching to the lower surface, right? So they are more confined to the upper surface only. So if the stomata will present more on the upper surface, there will be the more rate of transpiration. The water will evaporate, more and more water will evaporate, right? So to minimize that, to balance that, more stomata are present on the lower surface, that is the abaxial epidermis, than the upper surface, that is the adaxial epidermis. Mesophyll. It is a tissue found in between the abaxial and the adaxial epidermises. So in between both the upper surface and the lower surface epidermis, there is a tissue that is present in between these two type of epidermis, which is called as mesophyll, mesophyll cells. Okay. So this tissue is differentiated into the palisade parenchyma and the spongy parenchyma. Very, very important, right? So two types of mesophyll cells are present. Okay, the upper, that is the adjacent to this upper epidermis. Okay, upper epidermis, there is a palisade parenchyma, which is more of cylindrical type, and it is having lot of chloroplast in them. Okay, the lower epidermis, they are having round structures, round cellular or the oval structures, which are called as spongy parenchyma, and they are having many air cavities in between. Whereas in the case of palisade parenchyma, there are no intercellular spaces. Those they are cylindrical shape and they are compact together. So no intercellular space are present in the case of palisade parenchyma. So the palisade parenchyma is composed of tall, compactly arranged cells, while the spongy parenchyma comprises round or loosely arranged cells possessing intercellular spaces. So many intercellular spaces are present, and mesophyll cell comprise the chloroplast and carry out the photosynthesis. So this mesophyll cells are having a lot of chloroplast in them, so they can perform the process of photosynthesis. Okay. Vascular system. If you talk about the vascular system, then vascular bundles that are found in the leaves are closed and conjoined. If you talk about the vascular bundles, right, which is present in the leaves, they are closed and conjoined type. Closed means no cambium is present in between the xylem and phloem, but they are conjoined, they are close together, and which are engirdled by a thick layers of bundle sheath cells. So it is having outer covering of the vascular bundle, it is a thick layer which is called as bundle sheath cells. So this is the description of the dorsiventral type of leaf, the leaf which is found in the dicot stem. So here you can see the structures, the upper epidermis, okay, and that is the adaxial epidermis, which is having a thick layer of the cuticle to minimize the rate of transpiration. You cannot see any stomata on the upper surface, but here you can see the stomata are present on the lower surface, right? Lower surface. And if you see, so this is about the epidermis layer, two epidermis, adaxial epidermis and the abaxial epidermis, right? Now, if you talk about the mesophyll cells, so here you can see the mesophyll cells are of two different types, palisade mesophyll cells and the spongy mesophyll cells. Okay. Now, the palisade mesophyll cells are more of cylindrical type, and they are having the minimum air spaces in between. But if you see this spongy mesophyll cells, then spongy mesophyll cells is having a lot of air cavities in between. Right? And you can also see the substomatal cavity, which help into exchange of gases. Now, if you see the vascular bundles, then the vascular bundles, okay, they are having the bundle sheath cells, and the vascular bundle, there is a xylem and phloem cells, and in between there is an absence of the cambium layer. Right? So this is the structure of the dorsiventral leaf, that is the leaf which is present in the dicot stem. Okay, so this is all about this session. In this session, we have completed the exercise part of the anatomy of the flowering plants. We will have another session to discuss some other exercise. So we will meet again. Till then, thank you very much for attending this session. Take care and bye.